and and if there's like no relationship between them, it just means that they're acting independently. But so to specify my research further, I have decided to work with 65 different gene alterations that have been discovered to be significant in liver cancer, discovered by a previous research by Cornelius Schultz. But so far, before my research, their interaction has not been identified yet, so that's what I was doing in my own research. Oh, and just in case, before I go into the next slide, there are 3 main types of gene mutations. So first, there is the mutation in which the gene sequence changes. This can go either way. This can either be disabling the gene, or it can also make the gene form some other product that leads into cancer. So it can go to favor both turning a proto-oncogene into an oncogene, or just disabling tumor suppressor gene. And there's also deletion, which means that the gene is deleted entirely. Usually this goes for tumor suppressor genes because the cancer doesn't like the presence of something that's trying to stop them. And and finally there's the amplification, which means that there are multiple copies made of a gene. Usually the cancer cell would have multiple copies of certain proto-oncogene like VEGF-A, so that its growth and division will be most promoted. Yeah, so here's my purpose again. So the purpose of my research was to study the relationship of 65 previously identified important genes, mutations in liver cancer. And here's some of my references. So yeah, first I have the Cornelius Schultz paper, which really helped me decide which direction I wanted to go in. And Mayo Clinic also provided a lot of nice data for me to learn about liver cancer in general. The WHO and the CDC were also quite helpful for me to learn how significant the liver cancer problem was. And yeah, a lot of these. And finally, Illumina was also, and Illumina and CBio Portal were actually quite significant for me to learn about each properties of the gene, that 65 genes that I was studying, along with gene cards as well. These were very helpful when I was doing my research. So first methods. So I ran the 65 genes in a website that's called CBio Portal, and then I used a mutually exclusivity tab in order to see their relationships really. So basically the reds would be amplification in this place, and then the greenish blacks would be mutations, and the blues would indicate the deletions. So these are the types of mutations that each gene undergoes in liver cancer, basically. And so I first tried to kind of show all of these, and then I use a mutual exclusivity tab to try to see the relationships more. So the methods among the resulting 1,080 relationships, which is basically just 65 times 64 divided by 2, just NCR. I sorted out the 190 statistically significant relationships with p-values below 0.05. I'm going to go into what this means a little more, but yeah, basically the p-value less than 0.05 is the standard probability value with which we try to see if our relationships were significant. And I had to do such a hypothesis test precisely because I was not I am not creating a census of all the liver cancer patients. That's kind of impossible because there's a million cancer patients. But when we want to see the relationship in terms of the 366 cancer patients, which is actually quite a significant number, that can actually be have their genes checked for what are mutations in the liver cancer. Then we can run a hypothesis test like this to see if these genes actually mean anything. And basically these p-values would mean that these genes would have a strong relationship for the whole population. So this is a sort of generalization using a statistical test. So basically this is how all of these fancy numbers were calculated. The log odds, first the single the log odds ratio would be used. And you might be wondering why I precisely use the log odds ratio instead of just a plain odds ratio. So I had to do that because for the mutually exclusive relationships, the number of the odds ratio would be like getting closer and closer to 0. And like as it gets closer, it's going to be hard to discern the different numbers. But like if I do an ln, then I can actually make, especially the good thing about ln is that like I can make, when AD divided by BC equals 1, it means that there's no relationship at all. And then like I can make that as my frame of reference as 0. And then like any positive value would be like tending towards co-occurrence, and then like any negative value would be tending towards mutually exclusive relationships. So that's why like I decided to use the log odds ratio instead of just the odds ratio. And it's going to lead into like the second part. But first of all, in this like log odds ratio, A means the number of patients in whom both genes are altered. B is like only the first gene altered. C is only the second gene altered, and like D is both genes are not altered. And then I use something called the Fisher exact p-value test. So like what is this? So it's basically a table, like it's basically a test that allows me to see how likely a given table is going to result. So like a given 2x2 table in this case. So basically what this test does is it calculates all the combinations of like 366 into like basically like 4 numbers that add up to it inside a table, and then like with a given like sort of table that I can observe like of a gene. Like for example, if I have like 1, 3, like 4, and then like 360 like for like one table, the Fisher exact test allows me to calculate how likely that table is going to form. And it gives me the exact probability because it calculates the probability of each discrete table. So I use this in favor of the chi-square test. Like it's actually like the chi-square independence test is sometimes used for, I know that the chi-square independence test is sometimes used for like sample sizes greater than 5, but like it's actually being less used in favor of the Fisher like exact test for sample sizes less than 1,000 because the Fisher like exact test like it gives some more like precise values for like cell sizes less than like 5, any cell size less than 5, because some of like my research, as you can see, like it included like cell sizes far less than 5, like 1, 0 for example, that would make the cell size really small. And also yeah, it allowed me to like instead of like trying to like do an approximation of the p-value using the chi-square, because it actually creates an error like that allows like the significant like the p-values to be less than the significant level always. And like to bypass the problem, I had to make the Fisher like I had to use the Fisher test in order to ensure that like the test is not just like seeing any sort of random table and thinking it's like important enough. So that's why I use the Fisher exact test. And this is kind of like a table that's used for the Fisher exact test, and this is the code that I use for the co-occurrence. I had to use a different code for mutually exclusive apparently because like it only calculates like one side of the p-value. Like this code only calculates like one, either like mutually exclusive or co-occurrence, and I couldn't use like two-sided test because it would give the p-value that's twice as high. So I had to specify a different code for both mutually exclusive and co-occurrent relationships. And yeah, here is the table excerpt from like p-value less than 0.01, but here's a black and white version of the table up to p-value relationships less than 0.05 if any of you would like to see. It's black and white, but like it may like help for you to like actually look at it further. So in case of my research, like the p-value like less than 0.01 will mean that I can reject the null hypothesis that there is no relationship between the genes, and that there is likely a significant relationship between these genes for all like liver cancer patients, basically. And then like some of the color codes, let me explain. Like the reds and greens, like they're basically the same as before. Like, and then like the blue is like all these like are basically indicating which type of mutation each gene has. And then like the yellows are the indications for strong co-occurrence relationships, and like this purple is an indication of a p-value that shows a strong mutually exclusive relationship. So results synopsis basically. From the data I found that there were 13 mutually exclusive gene alteration relationships and 177 co-occurring gene alteration relationships that are very significant. So 64 of the total 65 genes were included in these significant gene pairs. I'll go over this a little more. And CTNNB1 was involved in a majority of the mutually exclusive relationships. And for the co-occurring relationships, the KMT2 group genes were really prominent. Especially in the p-values less than 0.01, these genes were practically dominating the table. Yeah, so, but there are other genes that I'll further go into, but this is the result synopsis. So, discussion. The prominence of CTNNB1, or beta-catenin 1 gene, in the results, for mutually exclusive relationships, actually helped to validate most of my other results, because the beta-catenin 1 gene is already known to be a very significant constituent of the WON pathway, which has been mentioned in the previous presentation by Helen, and also in other scholastic papers as well, to be a very important driver of liver cancer, and is associated with a lot of other genes in the major pathway of liver cancer. So the fact that it was present in a lot of the mutually exclusive relationships I found actually validated my research, retroactively in a way. But since 177 out of the 190 relationships were still co-occurring relationships, I felt that I had to find some of these and then try to explain some of them. And also, yeah, ACVR2A, it's something happened with that gene. That was the one that was not included in the significant relationships. I'll go into that a little bit further, just a little bit later. And the genes that were especially frequently found in co-occurrence relationships, as I mentioned, were the KMT2 family proteins, which are the lysine methyltransferase 2 families, especially the KMT2B, KMT2C, KMT2D, and KMT2E. And the substantial involvement of these genes in co-occurring relationships actually meant, actually suggested an epigenetic cause of liver cancer, because the KMT2 gene proteins would normally be associated with methylation of the histone, and basically what this would do is that it'll coil up a target gene and cause it to be less expressed in general. So the fact that these epigenetic regulators were mutated along with some other genes, and this was tied with the other mutations of the genes, basically the fact that KMT2 mutation, gene mutations having to occur with other mutations of other genes actually suggested that first some sort of epigenetic regulation has to be dysregulated for liver cancer, which actually leads into a potential drug research, because currently the new trend in liver cancer is basically scientists are trying to find different epigenetic treatments for liver cancer, and possibly this sort of finding could lead into additional research into let's say gene methylation and histone methylation. And ACVR2A didn't really form a statistically significant relationship with other genes, indicating that it does not really interact with other genes. So why is this really important if it's not interacting with any other genes? It's still important because that shows that it's independent from all the other gene mutations. And since it's already known to be a significant constituent that's driving liver cancer, that just means that it's a gene that's not associated with most of the other genes, and then it drives liver cancer independently on a pathway of its own. So it's actually quite a plausible hypothesis, because the ACVR2A is known to be involved in a pathway that's called activins. So it's another gene superfamily that doesn't include most of the 64 genes, and so this would suggest that basically it's ACVR2A mutation is driving the liver cancer in a completely different way than the other 64 genes are doing. So this actually was an important finding, even though statistically it was not an important finding. And it's also interesting that none of the deleted genes were among the dominant genes I found in the LMO2 slice. So there were not a lot of deleted genes forming a strong co-occurrence or mutually exclusive relationships where it's it's kind of likely that so it's kind of suggesting that the tumor suppressor genes are acting mostly independently in order to stop liver cancer, and their deletion is actually acting quite independent from a lot of the other dysfunctions of the genes in the liver, which is quite interesting to find. So, although future research is kind of necessary to further try to learn about these genes, possibly options indicate actually trying to do a pair CRISPR-Cas sort of experiment in order to knock out those genes that were deleted in liver cancer or mutated in liver cancer, for example, and then trying to see if these genes actually have a combined effect in driving liver cancer, and some other stuff like that, possibly adding a lentiviral vector also to simulate amplification of a gene. Although those future experiments might be needed, so far we're still thinking that some of these findings could definitely go into a potential new direction for drug research, especially ACVR2A, for example. It has not been studied for a major drug research in liver cancer yet, and my research is showing that possibly targeting that would reveal an entire potentially new field of drugs that can be created in order to treat liver cancer, as well as the KMT2 group proteins. Those are sort of new new directions for us to go in as well. So conclusion, presently I've identified the 190 significant relationships in the liver, liver cancer, all the genes that have been mutated, and CTMNB1 or the beta-catenin 1 gene formed the 13, majority of the 13 mutually exclusive relationships, which validated its importance in the Wnt pathway and retroactively in the formation of the liver cancer. And the genes that were found in the vast majority of the co-occurrence relationships were the KMT2 family proteins, and these other proteins, but mostly the most important one was the KMT2 family proteins. So it really sort of brings a new sort of light on the research of epigenetics in liver cancer, because KMT2s are important epigenetic regulators, and and also ACVR2A appears to act independently from most of the other genes. So that's definitely a potential marker to look for when we're trying to find a new drug treatment for liver cancer. And although more research is needed, these findings could lead into a potentially new area for drug research and aiding the therapeutic targeting of liver cancer. So that concludes my presentation. Do you by chance have any questions? Is there any kind of questions? Hello. Yeah.
How would you define epigenetics?
Epigenetics.
Yeah.
So epigenetics is basically all the things that affect the gene, but that doesn't come from necessarily the gene sequence. So for example, epigenetics is different from, let's say, mutation, amplification or deletion of a gene. It's some outside factor that, although the gene is completely there, it just activates or inactivates it. So for example, a gene methylation would cause, a cytosine methylation, for example, would cause the gene to not be expressed. Or as I was going over a little bit earlier, histone methylation of a certain gene can also cause the gene to tie around, pull around more tightly around the histone and cause it to be not expressed as much. So these things would be examples of epigenetics. Yeah.
Cool. Thank you.
It's actually kind of cool that these things can actually be transmitted from mother to son, for example. So it's actually a cool area to look at in epigenetics, how epigenetics is inherited from generation to generation. But yeah, that's a different area in my research.
So just going with the epigenetics,
Okay.
the theme. So when you say mother to son, so you're born with certain genes, but if those genes are not expressed, let's say your parents are both really tall, but then you could be really short. You're not born with the tall genes, or your tall genes are not expressed. Is that epigenetics?
Wait, what are you?
So you said, when you mentioned epigenetics is when genes are not expressed.
Yeah.
So, and you mentioned.
Or overexpressed, it could go either ways.
So let's say you're born from both parents who are super tall,
Yeah.
but you are really short.
Yeah.
So is that part of epigenetics, where your genes are not, their 12 genes are not expressed in you? Is that what it is?
It could be epigenetics, like, I mean, something might have happened that, like, some outside factor could have caused, like, I don't know, like, let's say the total, like, growth factor gene to not be expressed. But
I see.
at the same time, though, like, there could be other factors as well, like, such as, like, there could be, like, jumping inheritance, like, for example, like, so, like, a phenotype that does not necessarily show up in the parent stage because it's recessive, like, can suddenly show up in, like, the son stage because, like, it's just a recessive gene. So, like, actually it could, like, tie potentially in with the epigenetics, but, like, it could be more like a genetic sort of, like, factor as well. So, like, yeah, it could go, like, sort of both ways. I hope it answers your question a little more. Hello. Any other questions, Chase?